Welcome back to the playlist on amino acid oxidation. In this video, we're going to talk about the catabolic pathways for both phenylalanine and tyrosine. And what you should find good about this video and these pathways is that they converge with each other. So if you learn the pathway for phenylalanine, you will have also learned the pathway for tyrosine as well. So that's a good thing. What I always want to, what I always want to keep in the back of your mind is this. We're assuming in these catabolic pathway videos, we're making the assumption that we're in the fasting state. So if we're in the fasting state, that's implying that we have low blood glucose, we have glucagon as the predominant pancreatic hormone in the blood, and so forth, right? And so the liver cells are going to be doing beta oxidation, right? That's how they're keeping their NADH and ATP levels adequate, right? And they're also going to be catabolizing proteins down to amino acids, and they're going to be taking amino acids and degrading them into either ketogenic products or glucogenic products. Okay? If we have a ketogenic product, that's going to imply that whatever it is getting degraded to acetyl-CoA ultimately, right? And the acetyl-CoA can then be used ultimately to make ketone bodies, right? Or in some cases, we can degrade things straight to acetoacetate or something like that, right? And that's still ketogenic because it's a ketone body, right? But the whole goal of getting something down to a ketogenic product is we can basically make it into a ketone body that can be released into the blood and used by peripheral tissues, right? If we have a glucogenic product, that implies that we can in some way shunt it into the latter part of the TCA cycle and form oxaloacetate, right? And then the oxaloacetate can move into gluconeogenesis and we can make glucose. And of course that glucose can be released by the liver into the blood, and that's part of what maintains blood glucose levels, right? At adequate levels, okay? So I just want to have you keeping that in the back of your mind, that that's what we're assuming in these videos, okay? So when we're in the fasting state, the majority of our energy now is coming from beta oxidation and amino acid oxidation. Okay. Now, in this video, we're going to first look at the catabolic pathway, and then we're going to come back and look at some of the disorders that go along with some of these enzymes if they're deficient. Okay, so our first enzyme is going to be phenylalanine hydroxylase. And in terms of phenylalanine catabolism, this is the committed step. Tyrosine cannot be dehydrated back to phenylalanine, so this is an irreversible process. And phenylalanine hydroxylase is going to be a tetrahydrobiopterin-dependent enzyme. And over here on the right side of the screen, we note the structure of tetrahydrobiopterin. That's what's right there. And notice how it's fairly similar to tetrahydrofolate. Of course, though, it lacks the para-aminobenzoic acid ring and then also the glutamates that would be attached to it. So it's smaller than tetrahydrofolate, but it still keeps that pterin ring character. Okay? And the mechanism of phenylalanine hydroxylase is going to be the same as all amino acid tetrahydrobiopterin dependent hydroxylases. If we look at molecular oxygen, right, one of the atoms of oxygen will get incorporated into a hydroxyl group on phenylalanine to make tyrosine. And that hydroxyl group is right here, right? That's one of the atoms of oxygen in molecular oxygen. However, if we note over here on the right, which is the tetrahydrobiopterin cycle, the other atom of oxygen gets incorporated into the tetrahydrobiopterin as a hydroxyl group, okay? And that generates something referred to as hydroxy tetrahydrobiopterin, okay? Now, the letter that's A right here that we see is basically our hydroxylase. In this case, it's phenylalanine hydroxylase. There's also other enzymes like tryptophan hydroxylase, tyrosine hydroxylase. They're all going to follow this same type of mechanism. And what's going to happen is you're going to hydroxylate tetrahydrobiopterin to make hydroxy tetrahydrobiopterin, right? But hydroxy tetrahydrobiopterin does no good to us. So what we're going to do is we're going to regenerate the tetrahydrobiopterin. And that's ultimately done through steps B and C right here. Step B is something referred to as hydroxy tetrahydrobiopterin dehydratase. And we end up generating the water, and that water is ultimately going to come from this hydroxyl group, right? So we're dehydrating it, but in the process of dehydrating, we're going to get this molecule, which is right here, and this is called dihydrobiopterin. And the way you recognize dihydrobiopterin versus tetrahydrobiopterin is dihydrobiopterin is going to have this system of 3 double bonds right here, okay? In other words, you lose 2 hydrogen atoms in the process. So one of the characteristics of dihydrobiopterin are the 3 double bonds. But notice in tetrahydrobiopterin, there's only 2. Okay, so we're going to take dihydrobiopterin and reduce it back to tetrahydrobiopterin. And this is either an NADPH or an NADH dependent reduction. And this reaction is reversible, but if we're loading up the system with dihydrobiopterin by Le Chatelier's principle, we're going to force it back to tetrahydrobiopterin, and other enzymes can use the tetrahydrobiopterin. So the other forms of it really aren't any use to us. The only thing we care about is tetrahydrobiopterin, and dihydrobiopterin reductase regenerates the tetrahydrobiopterin. Okay, so now at this point, if I can find my mouse, we've generated tyrosine, and I want to point your attention to that this is a picture from Leininger's 6th edition of Biochemistry, and as we note from this reaction, it's not entirely correct. And what they're assuming in this reaction is they're assuming that you know the cycle right here. They're assuming that you know the tetrahydrobiopterin cycle. So we put in oxygen and we get out of water, but the water is not generated through phenylalanine hydroxylase. It's actually generated through hydroxy tetrahydrobiopterin dehydratase, right? Okay, so I just wanted to make that perfectly clear. This is not absolutely correct, but what they're doing is they're taking into account multiple enzymes. Okay, so now we have tyrosine, and tyrosine is going to react with an aminotransferase or a transaminase. And we note on tyrosine we have this alpha amine right here, right? We have the alpha amine, and we know that if we put it in contact with a transaminase and we have alpha-ketoglutarate present, we'll have a net substitution between the amine and the carbonyl, right? And so we should expect to get out glutamate, assuming we put in alpha-ketoglutarate. And what we're going to get out in the process is para-hydroxyphenylpyruvate. Now, the pyruvyl group, which I'll do in green, the pyruvyl group is this. And if you were to actually take off that para-hydroxyphenyl group, you would actually, and replace it with a hydrogen, you'd actually have pyruvate. So that's why it's a pyruvyl group. And so the reason it's para-hydroxy is because across that benzene ring, the hydroxyl group is para to the pyruvyl group. So it's para-hydroxyphenylpyruvate. Okay. Now, one thing you need to be aware of is when we're dealing with these aromatic rings, they're especially stable. We're going to find this also when we do tryptophan catabolism. Recall that tryptophan has an indole ring, which is actually even more stable than the benzene ring. Okay? And because the aromatic ring is so stable, we're really going to have to work it up in energy in order to get it to open up. So what we're going to have to do is react it with dioxygenases. And one of the things we have to understand about oxygen, molecular oxygen, is that it's an incredibly reactive species. In fact, if you go throughout Biochemistry 1 and 2, believe it or not, it's actually one of the most reactive species that we'll come across. And so it's high energy, and so what we can do is we can use that high energy and that instability and that reactiveness to react other molecules that are fairly stable. And ultimately what we're going to do in the next couple steps is we're going to use 2 dioxygenases, and the added energy we get from the molecular oxygen is going to open up the ring. Okay, and I want to make that perfectly clear. You're really going to have to work this benzene ring up in energy in order to cleave it apart, and that's ultimately what we're going to do. Okay, now think back to your organic chemistry 1 and 2. Okay, let me ask you a question. Did you ever see any reactions in those classes where you completely destroyed aromaticity? And the answer is no, you never completely destroyed aromaticity. You may have gotten benzene to react, right? You generated a sigma complex or a Meisenheimer complex or whatnot, but you always regenerated aromaticity. Well, in biochemistry, we can actually use the high energy power of molecular oxygen to actually cleave apart the ring. And you never saw a reaction like that in organic 1 or 2. But this is different. We're going to use molecular oxygen to open up the benzene ring. And the first step, which is a committed step, is catalyzed by para-hydroxyphenylpyruvate dioxygenase. And effectively what's going to happen in this is we're going to lose carbon dioxide. And I want to point your attention to the carbon dioxide that's going to leave. It's this carboxyl group right here. That's the carbon dioxide that we see right here leaving after we perform the dioxygenation. Okay? Another interesting thing about this reaction is we actually get a net carbon migration. So this whole group, and I'll eliminate the carbon dioxide for now. Actually, let me just put it all in there. this group is ultimately going to migrate onto this carbon right here. So in para-hydroxyphenylpyruvate dioxygenase, we get a net carbon migration. And of course, this is dioxygenase. So both atoms of molecular oxygen are going to get incorporated into the molecule. Okay, and that's going to give us something called homogentisate. And homogentisate is an interesting molecule. We'll do a little bit of an aside here from this pathway. And it's worth mentioning that homogentisate in other organisms has a life of its own. In other organisms, they can use homogentisate in order to biosynthesize plastoquinol, which is basically a coenzyme that's used in a photosynthetic animal's electron transport chain in the chloroplast, and they can also use it to synthesize two kinds of vitamin E's, and those are tocopherols and tocotrienols. Okay, so it does have a life of its own outside of this pathway in other organisms. So other organisms can catabolize phenylalanine and tyrosine down to this point, and then they can siphon off homogentisate into other biosynthetic pathways. So I just thought that was worth mentioning. Okay. Then homogentisate is going to get its aromatic ring completely cleaved apart. And this is catalyzed by homogentisate 1,2-dioxygenase. Okay? Again, it's a dioxygenase, so we should expect both atoms of oxygen to get incorporated into the molecule. And what we generate is something called malleate acetoacetate. Now, one thing I want to show you is that I'm going to draw the structure of maleate. So this is going to be maleate. Okay? Here is maleate. Oops, do this. Here is the structure of maleate. Now notice that structure is awfully similar to another molecule that we've seen, and I'll do that one in purple. And the molecule to which it's similar is fumarate, right? It's similar to fumarate. Well, what's the difference between these two molecules? Well, in maleate, which is shown in red, the double bond has a configuration of Z, right? Both of the highest priority groups are on the same side of the double bond. In other words, you could say those two carboxyl groups are cis to each other, right? But in fumarate, right, the double bond has E configuration, right? Because the two carboxyl groups, which are both highest priority, are on opposite sides of the double bond. Okay, so maleate essentially is the geometric isomer of fumarate, right? And so what we generate with homogentisate dioxygenase is we generate malleate acetoacetate. Okay, now notice that again it contains a malleate group, right? And if I was to circle the malleate group, it would be this. Right, this is the malleate group, and notice that it does contain a Z configuration alkene, right? But what the next enzyme is going to do is it's going to prepare the molecule for cleavage. And what it's going to do is it's going to isomerize that double bond. And so what you should see in the next molecule, I'll do this in orange, what you should see is a fumaryl group instead of a malleate group. Right? You see a fumaryl group. And you can probably guess where we're going with this. Usually when we generate something that's similar to fumarate, we're usually going to cleave it off and we'll basically have fumarate left, right? And so that reaction was catalyzed by malleate acetoacetate isomerase. But now we have fumaryl acetoacetate, so we have another enzyme called fumaryl acetoacetate or fumaryl acetoacetate hydrolase, and this is going to hydrolyze off fumarate from the remaining acetoacetate carbon skeleton. Now let's think about this for a second. Okay, what can the fumarate do? Well, the fumarate can go into the TCA cycle, right? This can go into the TCA cycle. And it would normally react with fumarate hydratase to give you malate, but ultimately that malate is going to react with malate dehydrogenase to give you oxaloacetate. And the oxaloacetate that's made can then be shunted into what? It can be shunted into gluconeogenesis for the synthesis of glucose, right? So what we've done through this enzyme fumaryl acetoacetate or fumaryl acetoacetate hydrolase is we've generated a glucogenic product, which is fumarate. And it's glucogenic because it can be shunted into glucose synthesis. But also in the process, we generate acetoacetate. Now, acetoacetate is a ketogenic product. Specifically, it's a ketone body, right? We actually don't have to do anything to it to make it into a ketone body. It already is, right? So we can say that this enzyme, fumarate acetoacetate, is both glucogenic and ketogenic. That's a rare occurrence. We really won't see a whole lot of that. And the acetoacetate that's made will be shunted out of the liver into the blood, and it will feed peripheral tissues. Now, what I want to make perfectly clear about this pathway is all of these enzymes up to this point are expressed in the liver. So this process of phenylalanine and tyrosine degradation is occurring in your liver during the fasting state. But this last enzyme that you see here, I don't even think they should have put it on here because it's a confusing thing. This enzyme right here, which they call 3-ketoacyl-CoA transferase, this one right here, another name that you might hear this enzyme referred to as is thiophorase. Let me also do it in purple. This is thiophorase. Okay. Now, what I meant before we actually do this reaction, what I want to make sure you understand is thiophorase or 3-ketoacyl-CoA transferase is not expressed in the liver. Okay, it's not expressed in the liver. So the acetoacetate that's generated by fumarate acetoacetate will leave the liver hepatocytes, go into the blood, and fuel other tissues. And one thing that can happen whenever you get acetoacetate is this enzyme, once acetoacetate gets into the peripheral tissue, thiophorase will catalyze the ligation of acetoacetate to the CoA, and then you can deal with the acetoacetyl-CoA. So notice what's happening. This part of the pathway, and I'll kind of section this off in black, this part of the pathway right here, this is occurring in a peripheral tissue, okay? So the acetoacetate leaves the liver, goes to the peripheral tissue like a muscle or the brain or whatever, and then that tissue ligates the acetoacetate to CoA. So I want to make that perfectly clear. Every other enzyme is in the liver, but this one is not. Okay, so the acetoacetate will get into the peripheral tissue, and 3-ketoacyl-CoA transferase, or thiophorase, will catalyze the ligation of acetoacetate to coenzyme A. And notice where the CoA donor is, right? The CoA donor is actually succinyl-CoA. In other words, that succinyl-CoA will have to be taken out of the TCA cycle, but the good thing is we get succinate back out of it, right? We get succinate back out. And of course, that can go back into the TCA cycle. Now, real briefly, let's think about acetoacetyl-CoA. Okay? Well, the acetoacetyl-CoA that's generated by the peripheral tissue can then be cleaved by thiolase. Right? So I'm actually surprised if they're going to put this reaction in there. I'm surprised they didn't do this. But this can react with thiolase, right? Or in the reverse direction, it's usually named as acetyl-CoA C-acetyltransferase. And what we should get out of this reaction are 2 acetyl-CoAs. And of course, those acetyl-CoAs can be shunted into the TCA cycle of the peripheral tissue. So what did we just see? Well, we saw that phenylalanine and tyrosine get degraded ultimately to fumarate and acetoacetate. Now, that reaction of fumarate acetoacetate is expressed in the liver. So the fumarate can go into the TCA cycle of the liver cell, right? And you can ultimately shunt it into gluconeogenesis to form glucose. And that glucose gets released into the blood and can fuel peripheral tissues like the brain and muscle. But the acetoacetate that's generated just directly leaves the liver cells and goes to a peripheral tissue where thiophorase, or 3-ketoacyl-CoA transferase, ligates acetoacetate to coenzyme A with the loss of succinate. And of course, the succinate goes back into the TCA cycle, but the acetoacetyl-CoA gets thiolized by coenzyme A to make 2 acetyl-CoAs, which go into the TCA cycle of the peripheral tissue. Okay, so the last 2 steps, 3-ketoacyl-CoA transferase and thiolase, those that we're talking about are not done in the liver. Those last parts are just peripheral tissue enzymatic reactions. Okay, so I hope this video gave you a little bit of intuition on phenylalanine and tyrosine catabolism. Now what I want to do is I want to shift gears a little bit. In the next video, what we're going to do is we're actually going to look at some of the imbalances that we have, specifically phenylketonuria. And actually what we have in this picture is on the left side of the reaction scheme, to the left of the enzymes, those are the disorders that are usually associated with deficiencies of those enzymes. And then on the right side, they're to the right. Okay, so for instance, a deficiency of phenylalanine hydroxylase would be called PKU, or phenylketonuria. Okay, and oftentimes what these deficiencies are caused by is there's some kind of mutation in the gene that codes for the mRNA, that codes for the protein. And so if you have a mutation in the DNA, it'll affect the amino acid sequence of the protein. And if you affect it enough to where you lose function of the enzyme, then you lose the reaction completely. So you have a mutation in the gene that messes up the function of the enzyme, so the enzyme doesn't work. So for instance, that would be one of the causes of phenylketonuria, a mutation. Okay. And so if you have a mutation in phenylalanine hydroxylase, you have this disorder. Even though you need phenylalanine to live, you don't want a whole lot of it. Okay, it's like my inorganic professor said, the dose makes the poison. Okay, you don't just want a ton of phenylalanine floating around the cell. Okay, you have to have a way to get rid of it, and as we'll find in the next video, there actually are auxiliary pathways to get rid of phenylalanine, but they're not near as efficient as this pathway. So in other words, if you have a deficiency of phenylalanine hydroxylase, you can't clear phenylalanine by this pathway. Okay, and not only that, you can't biosynthesize tyrosine. Okay, notice that this pathway is technically a biosynthesis of tyrosine from phenylalanine, assuming you start with phenylalanine. So not only would you not be able to make tyrosine, which would give you somewhat of a deficiency if you're not getting it from the diet, but you also can't get rid of phenylalanine, and it's toxic in large amounts. Okay, and because it's toxic and it can accumulate because of this, people with phenylketonuria have to worry about phenylalanine in the diet. Now, one of the problems with this is, of course, you have to have phenylalanine to live, right? There are a lot of proteins or enzymes or whatever in your body that have to have it for their three-dimensional structure, or we could even say their primary structure, right? And if they don't have phenylalanine, the protein doesn't get made. So you have to have some of it, but you can't have a lot of it. So it's one of those things that you sort of have to titrate in your diet. And so you have to worry about eating foods that are especially high in phenylalanine. And one of the things that's of medical importance is I want to look at aspartame. And if you'll give me just a minute to draw aspartame, okay, and of course I'm drawing it from N-terminus to C-terminus, okay, it's basically a dimer of aspartate and phenylalanine. Okay, so the first amino acid in this very small peptide is aspartate, okay? And the second amino acid is going to be phenylalanine, okay? Second amino acid is phenylalanine. But this is an unusual peptide in that it ends in a methyl ester. So it ends, instead of ending in a carboxylate C-terminus, the C-terminus actually has a methyl as making an ester, right? And this molecule right here, this is aspartame, okay? Now, one thing I want to do is I want to think about the complete hydrolysis products of this molecule, right? What happens when we hydrolyze it completely? And you have to keep in mind, how many carboxylic acid derivative bonds do we have? Well, number one, we have a bond right here. There's a carboxylic acid derivative, right? That's an amide linkage, right? And you have to keep in mind, there are proteases or peptidases that will degrade that bond, right? They'll break that bond in a nucleophilic acyl substitution, and probably it's going to be a serine hydrolase that does it, right? And then you also have this carboxylic acid derivative bond, right? There's the bond that's part of the ester. And although there aren't any enzymes that degrade that, the actual cleavage of that bond is spontaneous. So let's think about the hydrolysis products of aspartame. Well, number one, you're going to get the first residue right here, and that's aspartate, right? That'll be your first hydrolysis product. And then, if you were to cleave this bond right here, you're going to get two products. You're going to get, number one, phenylalanine, and you're also going to get methanol, which we're not really concerned with here. Okay, although you don't want a lot of methanol. But the whole point is, if you were to, let's say you had a deficiency of phenylalanine hydroxylase. In other words, you had phenylketonuria, right? What happens theoretically for every single molecule of aspartame that you ingest? Well, theoretically, for every molecule of aspartame that you ingest, you're going to get one phenylalanine. So in other words, if you were to ingest 1,000 molecules of aspartame, you would get 1,000 molecules of phenylalanine. And let me ask you this. Can you think of a drink that has a lot of aspartame? And the answer is something that you're probably accustomed to hearing of, and those are diet drinks. A lot of diet drinks on the market today contain aspartame. And so one of the things that is required by the FDA is they require that on the label of the drink, they say contains it contains phenylalanine, and of course they spell out phenylalanine. But it says contains phenylalanine, and that's because phenylalanine is part of aspartame. Okay? If you were to do the complete hydrolysis of aspartame, you'd get phenylalanine, along with aspartate and methanol. And of course, if you have this disorder and you're drinking a lot of aspartame, you're also, so too, drinking a lot of phenylalanine. And that phenylalanine builds up. Right? And although you need phenylalanine to live, you can't have too much of it. And that's one of the sort of downsides of this disorder. You have to have it to live, but you can't do too much of it. There has to be some sort of happy medium. But either way, it's just a terrible disease, and you're going to have problems down the road. And in fact, if they don't catch it soon after birth, consumption of phenylalanine will cause things like mental retardation, and throughout life it can cause things like seizures. So it's just a terrible thing, terrible atrocity. Okay, and there are certainly other disorders associated with this pathway. It turns out that disorders of this pathway are actually fairly common. Another one is called alkaptonuria, and if you have a deficiency of homogentisate 1,2-dioxygenase, you get alkaptonuria, and what essentially happens is homogentisate builds up. And you get things like early onset arthritis and all sorts of nasty things like that. And certainly you can see that we also have tyrosinemia. Tyrosinemia 2 is a deficiency of tyrosine transaminase. Tyrosinemia 3 is a deficiency of para-hydroxyphenylpyruvate dioxygenase. And the worst of the tyrosinemias is number 1, and that's a deficiency of fumaryl acetoacetate hydrolase. Okay? It's a deficiency of fumaryl acetoacetate hydrolase. Okay? And of course, in the process, if you were deficient in any of these enzymes, you have to keep in mind that you're going to lose out on the fumarate and you're going to lose out on the acetoacetate. So your energy yield goes down with any deficiency of any of these enzymes. Okay? And just keep in mind, they're most likely caused by mutations in the gene that codes for the enzyme, and that messes up the protein's primary structure, which is the sequence of amino acids, and therefore if you change that, you change the three-dimensional structure. Okay, so I hope this video gave you a little bit of intuition on this catabolic pathway and some of the disorders that are associated with it. And if you're going on to med school, these are things that you have to know. See you in the next video.